Okay, so primarily what we are shooting for, we are trying to kill the cancer cells. Yes, that is our target. So cancer cells, along with the normal cells, they are there and progressing. Only difference between cancer cells and normal cells having a mitosis is cancer cells are faster dividing. So can the chemotherapeutic drugs differentiate these degrees of cell cycle division? Absolutely not. So that is the reason if you are if anybody is exposed to chemotherapeutic agents, they also have loss of hair, uh, like membrane of the mucosal membrane, things like that, which are faster dividing cells. So that's the major problem with non-selective chemotherapeutic drugs. But so far, we are trying to invent drugs which can be more selective towards the cancer cells. But so far, it is still in the progress. What we have, what we are going to talk about today are the two fundamentally used drugs, like classes of drugs. One of them are the alkylating agents. These agents actually were used during the World War II to kill people. Mustard gas, still now in several parts of the world, some people actually use those mustard gas to kill people. Like kill people in the sense like people don't die just like that. Their DNAs gets mutated, means alkylated. So they have mutated DNA, have a severe cancer, and eventually die. So that's the whole uh, point of targeting alkylating agents or talking about alkylating agents. These are some of the learning objectives, what we are going to learn today. Like I am asking you to read through so that you are preparing yourself to tackle questions which are going to come from here. So primary thing, what am I going to talk about are what are the characteristics of the common characteristics of the alkylating agents. Alkylating agents, as I was mentioning, they alkylate DNA in a specific position. I will come to that in a minute. But what do they do? This is fundamental. They covalently attach. So I already put highlights on that. They covalently attach on the DNA. And these, because of this covalent attachment, we know that covalent bonds don't break just like that. So they are irreversible. Because of that, it causes permanent damage and kill the cancer cells. But again, can they differentiate between the cancer cells, which are fast dividing cells, and the other fast dividing cells in the patients? No, they have they have no selectivity like that. That's why when they are exposed to these alkylating agents, people tends to lose hairs and nails and things like that. Now, again, what I said by beginning the lecture, I said they stop cell cycle division. So they have cell cycle specificity. Yes, all these important things which you do need to remember in order to tackle not only in the class, but for your NAPLAX exam, that are already highlighted. So alkylating agents are most cytotoxic to all the rapidly dividing cells. As I was saying, that they target express they do not differentiate between the normal fast dividing cells and the cancer fast dividing cells. It affects cells in all the phase. The primary important phase is the S phase, which is the synthesis phase. I will talk to you in a minute. Polynucleotides are more susceptible to alkylation in their unpaired stages. So what we are talking about, if I am talking about that we are having a rapidly dividing cell when it is having a like when new chains are synthesizing for synthesizing replication of the DNA, that time the DNA two strands, they are not bind together yet. So that means those nucleotides are vulnerable. At that point, if the alkylation happens, then that nucleotide, which is supposed to bind with the other nucleotide of other chain, doesn't happen. There is a mislink happen. So what is the mislink? We will talk about in a minute. So this is the S phase, which is the DNA synthesis phase, where this is most vulnerable to alkylating agents. What is happening? Usually we see that cysteine binds with guanine with the 3 bonds, right? And A binds with T, thymine. Adenine binds with T, means thymine, in 2 different strands. But because of this mismatch, what happens? Can we see this at the bottom? If we cannot see it, we should be able to see it when you are going to look at your slide. So here we are seeing that guanine is binding with cytosine instead of like guanine is binding with thymine. Instead of cytosine, it is binding with thymine. That is the mismatch. Because of this mismatch, there are mechanisms in replication where it can repair mismatch, but this kind of mismatch cannot be repaired. So it causes permanent damage to the cell cycle division. Yeah. It's not there. Okay. I can put an updated slide. Okay. And it is there also in another slide. It binds covalently with the alkyl group binds covalently with the guanine. So because it binds covalently, one of the donating electron is already occupied. So it cannot now form 3 double bonds. It can only form 2 double bonds. So that's why instead of binding to cysteine, it binds to thymine. So it's a complete mismatch. Normal DNA helical pair are supposed to have binding between guanine and cysteine, but in this case, because this alkylating agents already bound to a certain position, it cannot bind to thymine anymore. It cannot bind to cysteine anymore. It will bind to thymine. So that is the abnormal base pairing. This is the abnormal base pairing. Make sure you highlight that. So abnormal base pairing between G and T. So cross-linking of DNA, ring cleavage. So basically what we are doing, we stop DNA replication. That means we stop DNA synthesis. If we stop DNA synthesis, that means cell won't divide anymore. We stop cell cycle division. That is the primary target of these alkylating agents. That means these chemotherapeutic agents, we are stopping cell cycle division. So where it is binding, it is extremely important for us to know that where it is binding. N7 of the guanine, that is the primary alkylation site. Okay. So it is binding to the N7. So it is already occupying this one. So it cannot bind to 15 anymore. It has only 2 bond availability. Okay. The common characteristics of all the drugs, what do they have? If they do need to have ability to reach tumor, that means they need to cross the tumor to cell, like cellular membrane of the tumor, right? So they need to have a certain lipophilicity, otherwise they won't cross the barrier. We know that from our general perspective in the 314 course, that where we were talking about lipophilicity is the primary criteria for a drug to cross the membrane barrier, including the blood-brain membrane. Okay. Now, these has to have, like, again, it is a repetition, like lipophilicity, ability to reach tumor. That means it needs to have cross the biological membrane. Then acid dissociation constant, that means it needs to, like we all know that if we are talking about pKa, acid dissociation constant needs to have a factor in the pK, like in the log P value, log P calculation, way going back to the 314, and stability in aqueous solution, sites of macromolar attack. That means the macromolecule which we are talking about, the DNA, it should be able to attack that particular macromolecule. So these are primary criteria. In the history, I'm not going to give you history lesson, but as I was mentioning that this compound or chemical was used during the nitrogen mustard as a nitrogen mustard gas during the World War II to kill people. And World War I, also World War II. And since then, several times, lot of tyrant leaders in the world, they have used it to their own people or to the other people. But in any way, this is a knowledge check right at this point to be able to see exactly what we have learned, and you can answer that. Pretty much it gives you hints for what kind of questions you are expecting from this portion. Now we are into the drugs. The first one are the nitrogen mustard. There are different varieties of that. These are the structures. If we are looking at the structures without paying too much detailed information, to like paying too much detailed attention towards the structures, what we can see that it has an ethyl chloro substitution, right? Nitrogen. So that's the primary essential features of any nitrogen mustard. Also, this is the compound cyclophosphamide. Please pay attention to that. I will come back. So nitrogen mustard, common features. It is tertiary amine. It needs to have the 2-trichloroethyl group that cause the bis trichloroethyl group. That is a fundamental and essential features of the nitrogen mustard. And aliphatic or aromatic substitution of the R, because it is a tertiary amine. So all these features are important, and I'm highlighting them so that when the question comes, you can tackle them. So basically, what is happening from this structure, it makes an intermediate that intermediate is known as aziridinium ion. So this is again fundamental. What is aziridinium ion? So if I give you this structure, or if I give you a question that what is the fundamental intermediate going through this mechanism of attacking the DNA, you should be able to tackle and answer aziridinium ion. Okay, what is aziridinium ion? To make it simpler, it is it's a triangular, like a like cyclic structure, three-membered cyclic structure with a nitrogen. And aziridinium, always ums come with the positive ion. Okay, so aziridinium is a positively charged species. This is extremely sensitive ion. That is the reason it can cause alkylation to the 7th position of the guanine. This is the 7th position. This is electron deficient. Nitrogen has lone pair of electron. It made the nitrogen to attack that electron deficient compound, which is the aziridinium ion, and cause a covalent bond formation. So that's the fundamental thing. Aziridinium ion, N7 guanine covalent bond formation. So nitrogen mustard, if I read through, highly electrophilic quaternary aziridinium ion formation. That is first step and fundamental. What is the second step? It attracts nucleophilic guanine N7, as I was explaining to this little drawing on the side. And then this cause two of these alkyl substitution to happen on the aziridinium ion. So basically it opens up and another guanine gets attached. So in between one alkylating agent and two guanine ion, like guanine adduct. So basically it is extended through the chain of one strand. If it does happen, what happens to the other complementary strand? The complementary strand, in order to bond form, it does not have this nitrogen available to make any bond formation. So instead of making 3 bond formation with the cytosine, now it forms 2 bonds with the thymine. So that is a mismatch completely. And this cannot be repaired by the DNA repair mechanism. Cyclophosphamide, I said I will come back. This is another extremely important statement. Cyclophosphamide is a prodrug. Okay? If we look at the structure, we easily should be able to identify, but I'm not going to bother you with too much structural information. Just for you to know, cyclophosphamide is a prodrug. Now, the alkylating agent pharmacology. Covalent modification of guanine molecule. We already talked about that so many times. Formation of cross-links of DNA strands, like DNA strands in this position, A DNA. So this forms this guanine to the cytosine. Like instead of cytosine, it forms to the thymine. So imidazole ring cleavage, disruption of the guanine base, and mispairing with thymine instead of cytosine, right? So this is the fundamental thing. What do we need to remember from here? again, the things which we already talked about and learned about, that N7 alkylation, because of that mispairing of guanine to guanine to thymine instead of guanine to cytosine, which is normal. Okay. Now, this is the knowledge check. Again, this is going to tell you what kind of questions you are going to expect from the material, what we just learned here. Okay. Now, next classes of drugs are cisplatin. Still, people use cisplatin. This is the first line of treatment when you will hear from Newman, Matt Newman, you will hear cisplatin is the first line of treatment for ovarian cancer. But even though it is long days passed away, this is the first line of treatment. Now, cisplatin or carboplatin or oxyplatin, what is their common? Platin, right? So it will have a platinum analog. So the fundamental thing that all these drugs in this class are platinum analogs. So these are the structure. What is the fundamental thing in these structures? I'm just going to tell you and going to show you. I already wrote the fundamental things. This is square planar. That means platinum in this state can only stay as a square planar. That means square planar means it's nothing but a flat structure. So what is the benefit of having a flat structure? It can intercalate in DNA. Means all those bases, they are aromatic. So it can easily stack on that and intercalate with the DNA. So that's the fundamental benefit of having like this platinum derivatives. Again, the structural features, fundamental, it's a square planar structure. Second things, it needs to have platinum because we are talking about platinum derivatives. Platinum is in a positive state. It will have leaving groups and it needs to have amines. These are the four fundamental features of platinum derivatives. When we are going into the pharmacology, what we are seeing that this platinum can bind like this. It will sit on top of a particular strand in such a way, because as I was saying, it's a flat structure. So it can easily get into the DNA double helical strand and binds with 7 position of the guanine. Again, the 7 position of the guanine is the most vulnerable. Why? Because that nitrogen is most available for donating electrons. That's the most basic nitrogen. If that is the case, those nitrogen pairs are available to form covalent bonds. How it's going to bond covalent bonds with platinum? Because platinum again is in positive state, electron deficient. What did we see for the alkylating agents? We saw aziridinium ion, which is again a positive core, which is attracting electron to come and bind. So if that is the case, here the platinum is also giving the same particular environment that it attracts the nitrogen lone pair of electron and binds. As soon as it binds, the other hand, and as I was saying, if we look at that, what did I say? It has a platinum positive and then two living groups, right? These are living groups. So they leave and binds with the guanine. So the structure became like this with two bases of guanine. If it does that, then what is the next possible answer? Instead of binding guanine with cytosine, again, it is going to bind with the thymine. So again, mismatched base pair between the strands. Yes? So moving on, it is again expressing what actually happens. Cationic platinum attracts adjacent nucleophiles. What is the adjacent nucleophiles? Guanine, nitrogen at the 7th position. Cis orientation of the displaced ligands cause to bind two guanines because it is cis. If it would have been trans, it would have bound only to one guanine. What is the difference between binding to two guanine instead of binding to one guanine? In one guanine, it is very easy for the DNA mismatch repair mechanism to kick off that. But if it binds to the two guanine, it is difficult because now it is a cyclic structure. It does not leave. So guanines are occupied with one position. Which one is that? N7. So instead of binding to cysteine, it now binds to thymine. The most common intrastrand occurs between diguanosine dinucleotide, right? Because it forms two guanine binding. Cross-linking results in altered base pairing. What is the altered base pairing? That is G2T instead of G2C. Moving on. The last class in this alkylating groups are DNA methylators. They are extremely sensitive compounds. I actually work with this kind of compounds, not with the DNA methylators, but diazomethane. We will talk about that in a minute. What it does, it is an extremely sensitive material to cause DNA alkylation. What it does, in situ, it makes diazonium, methyl diazonium ion. So again, this is another thing you need to remember. One was the aziridinium ion, other was the diazonium ion, diazonium salt, right? So this diazonium cause, again, this is a nitrogen, it's a deficient nitrogen species. It caused to release the nitrogen gas and makes an electron deficient carbon. That electron deficient carbon gets attached with the N7 of the guanine and thereby alkylate the guanine. Again, next step, if it alkylates guanine, the base mismatch will occur. Instead of GC, it will be GT. Okay. Procarbazine pharmacology mechanism of action is unclear, and inhibition of DNA and RNA protein both. So these are the ones which I'm talking about, this procarbazine, which are extremely highly sensitive methylating agent, which generates methyl diazonium salt in situ. Now, again, the knowledge question, knowledge check portion is here for you to understand. Next portion, what we are going to talk about, this is one of the fundamental classes of oncology drugs are commonly used, which is learning objectives is there, which is dihydrofolate reductase, DHFR. In order for us to understand the dihydrofolate reductase, I listed some of the structures I am asking you strongly to go back and revisit the structural information of the bases. It's nothing but to go back and remembering guanine, adenosine, and all these things, right? Okay. What are the different classes? Anti-metabolites. First one is the folate antagonist. Second one is purine and analogs, and the third one is pyrimidine. So primarily in this class, clearly saying that these classes of drug has to be flat. They intercalate the DNA and thereby cross-strand bind. So because of that cross-strand binding, DNA after replication do not separate. That means further replication is stopped. DNA replicate, then bind, then again separate, form another strand of DNA, which is replication. But what we are doing, we are stopping replication by making the two DNA strands as strong as possible so it cannot dissociate anymore. So that's the fundamental mechanism. There are all these compounds of this, and these are the structures. If you look at the structures, again, the fundamental features you can look at would be the aromaticity of the compounds. That means flatness. Structurally, these agents mimic the naturally occurring nucleotide cousins, means either the purines, pyrimidines, or relatively similar purines, like relatively similar pyrimidine derivative. That means dihydrofolate reductase, methotrexate. We will see that. Inhibiting enzymes involve nucleotide synthesis. So it stops nucleotide synthesis. Inhibiting enzymes involve the DNA replication, replacing naturally occurring nucleotides in DNA that is actively being replicated. So basically, as it intercalates, instead of the regularly occurring bases, these now participate. So that means no longer it can replicate. As I was saying, that it forms a cross-chain bond formation. Okay. This chart gives you a summary of information of different of these drugs and which enzyme it affects. It's a very, very concise summary slides for you to remember very easily. I will put an asterisk beside it so that we would know that all these compounds, what enzymes do they block? Again, they have cell specificity for cell cycle inhibitor S phase. Why? Because S is the DNA synthesis phase. What it does do is stops replication. Again, this is the knowledge check explaining that what kind of questions you are going to expect from these portions, this small portion. First one is the folic acid analogs. The fundamental thing is, what is folic acid? Folic acid is vitamin B9. So you need to know that what is folic acid. Then important cofactors. What is folic acid important for? Important cofactors for production of purines and pyrimidines, reduced by dihydrofolate reductase. So it is a mechanism where it needs to get reduced, dihydrofolate reductase. So these compounds block dihydrofolate reductase. What is the structural features? It has a pteridine ring. That's the way you pronounce. T does not pronounce. It starts with T, pteridine ring, even though it has a silent P. Pteridine ring is this. I'm asking you to remember this ring because I can put this ring and ask you to identify what kind of ring is this. Is this a quinoline? It's a quinazoline, pteridine, what? So this is a pteridine ring, one. Second thing, para-aminobenzoic acid and glutamic acid. These are the three fundamental features. So I wrote the structure, but that doesn't stop me for not to give you the names of this. So it will be easier for you to remember. Pteridine ring, para-aminobenzoic and glutamic acid. Now, if we move on, first one is the methotrexate. This is C4 amino substituent, enhances the basic character of the N1. So it has a pteridine ring. Why? Because it is a folic acid analog. It replaces folic acid. Instead, it brings methotrexate. So cause that to incorporate into the DNA synthesis. So it stops, enhances the basic, here the enhances the basic character of the N1, promoting the protonation over N5 that orients the molecule in such a way that it cannot be reduced like folic acid. So what is the cause of stopping the reduction of folic acid, which is a natural mechanism for the synthesis of the nucleotides? What is the reason? Because it has some substituent which is causing it to stop the reduction process. You do not need to remember N1 or N5, but what I'm saying, like basically structural modification cause it not to do the folic acid reduction. That means it's blocking the folic acid reductase enzyme. Second one is the hemetrixase. What is the benefit of that? It changes the pteridine ring to purine ring. Instead of the pteridine ring, it has now a purine ring. So it completely changed the structural features. So no longer it is available to reduce the reduce the folic acid to its complementary reduced portion, because the dihydro, so basically it's stopping the dihydrofolate reductase enzyme. Coming back to the pharmacology, methotrexate dihydrofolate reductase enzyme inhibitor. Also, it is thymidylate synthase inhibitor. Two enzymes, dihydrofolate reductase and thymidylate synthase. Those two enzymes are blocked. What is the benefit of, what is the mechanism of methotrexate? This is also blocks the thymidylate synthase and dihydrofolate reductase. We already saw dihydrofolate reductase, how it blocks, because instead of the pteridine ring, it has a purine ring now, right? So whole molecular structure is changed. Folic acid analogs, inhibition of the dihydrofolate reductase reduces the intracellular level of tetrahydrofolate. This is the reduced product, which is tetrahydrofolate. Reduction of that tetrahydrofolate is necessary for the synthesis of dump to thymidylate, synthesis of purines A and G, synthesis of pyrimidines, and amino acids serine and methionine. So what I am asking you to remember, dihydrofolate reductase inhibitor, methotrexate, pemetrexate. What it does stop production of the tetrahydrofolate, THF. These tetrahydrofolate are necessary constituent to synthesize the bases all these bases, the pyrimidines and the purines. So it stops DNA synthesis. If it stops synthesis of the nucleotide, it will stop DNA synthesis. So pemetrexed, what is the mechanism? It's converting DUMP to DTMP. I expressed what are the full expression of DUMP and DTMP. You don't need to remember that, but in case you have inquisitiveness, you can still look at that. Okay, so fundamentally, after studying this material, what you have learned, that is going to give you hint what kind of questions you are expecting. The last two groups, which I'm going to get done in a brisk, purine analog. So that means it says that it resembles purine. So if it resembles purine, it will cause mismatch, like it will cause a really strain in the DNA synthesis, because you need purine. No longer you have purine available. Purine-like structure. This chemical structure mimics the naturally occurring purine. Drugs are converted to their nucleotide form and incorporated in the DNA. So instead of purine, you have these modified structure incorporated in the DNA. How does it work? One of the drug is cytarabine. How does it work? It's a cytosine analog. It's telling you what kind of analog is that. So this is the thing you need to remember. Once you have cytosine analog, no longer the regular synthesis of DNA occurs. What is the regular synthesis? Once inside the tumor cell, it converted to ARA-CTP. Instead of CTP, it converts to ARA-CTP. ARA-CTP is described in here. Arabinosyl, furanosyl, cytosine, triphosphate. We don't need to know that, but this is what it is. Okay. Okay. The pyrimidine analog, the last one, is 5-fluorouracil. This is one of the most important drugs in cancer chemotherapy. 5-fluorouracil. It resembles a pyrimidine analog, and structure is like this. What it does, it causes, again, instead of a pyrimidine analog, you have this 5-fluorouracil which sits in the DNA, which cause some kind of perplexed action for the DNA synthesis to occur. So it stops. You no longer have pyrimidine. Instead, you have a different type molecule which cause to stop the DNA synthesis. Pyrimidine analogs are the capecitabine. Oral product is, this is really important statement. Oral product of 5-FU, which is 5-fluorouracil, designed to be activated selectively by tumor cells. So 5-fluorouracil is an oral prodrug. Are we clear? That is the whole fundamental thing. Other things I'm not asking too much for you to remember. Now, the two marketed products differ only in the mechanism of activation for the 5-fluoro-DUMP, DUMP. So what is that? 5-fluorouracil is a base, requires conversion to the deoxyribonucleotide via ornate phosphoribosyl transferase. You do not need to know that. Only thing is 5-fluorouracil, the first line, 5-fluorouracil requires conversion to deoxyribonucleotide for the 5-fluorouracil. For the captivin, it is a false cytidine nucleoside. So basically, it is giving you a completely different cytidine structure. As we were talking about it before, that it caused the arabinocytosine triphosphate to form. So these are the two fundamental mechanistic difference. You do need to remember that. Now, if we go to the last thing, this is the knowledge check, what you have learned in these pyrimidine analogs. So what we have learned, we have 2 minutes to recollect. What we have learned is we are targeting cell cycle division. How? Primarily targeting the S phase of the mitosis, which is DNA synthesis. How? We are using the alkylating agents. It's changing the bases, it's alkylating. So changing the bases and causing it to mismatch. Instead of G and C, it's causing it to connect with G and T. So it's a complete mismatch. You won't have DNA synthesis anymore. It will stop. But the other guys, anti-metabolites, what it does, they mimic the nucleotides. That means purines and pyrimidine analogs. They mimic that. So if they mimic, instead of the regular pyrimidine, like regular nucleotide, you have now this substituted nucleotide which is sitting in place that will again stop the DNA synthesis. So that's the whole fundamental concept. Stopping DNA synthesis, thereby inhibiting cell cycle division, where it's targeting S phase. For the alkylating agents, all of them, it is alkylating the N7 position of the guanine. Because of that, guanine no longer binds with cytosine, it binds with thymine. Fundamental. And the difference is for the dihydrofolate reductase, the purine analogs, and the dihydrofolate reductase inhibitor. So it stops DNA synthesis. And the compounds which mimic purine and compounds which mimic pyrimidine. So it is all recorded. I know like you did not pay attention to listen to any of this because I see you are like concentrating on your exam material. But again,